Hi, let's talk flu virus replication cycle. So, I want you to be able to walk me through this entire cycle. But the beautiful thing is, it's got a lot in common with the other viruses we've studied, HIV and the coronavirus. So, here we go. Listed down at the bottom are all the steps, 1 through 6. And yeah, we'll go through them in not much detail now. And then we'll go through them in more detail in some of the later slides so you can sort of fill this in. So when you're taking notes, I want you to get a good picture of the entire overview of the process. How does this work? So here we go. So step one is going to be attachment and entry. And so attachment happens when one of those two proteins at the surface of the virus, so the hemagglutinin or the neuraminidase, in this case it's going to be the hemagglutinin, when it binds its receptor, then it's going to enter. Now, the neat thing about the influenza hemagglutinin is that it does not bind a protein directly. It binds to the sugars on the outside of a protein. So it binds to a thing called sialic acid, or an older name for that would be neuraminic acid. And this is going to be on the outside of nearly every protein on the outside surface of a cell. And so once a flu virus gets to the right place, it is going to be able to enter, and it can use, yeah, any protein it wants, theoretically. Uh, yeah, to get in. All right, so it's going to enter, and that process is going to look real familiar. So it's going to get pulled in via something like a clathrin-coated pit, and we're going to acidify that vesicle as if we were sending it to the lysosome, because we are, because cells are super predictable like that. And so there are going to be histidine amino acids in the stalk of the hemagglutinin protein, the HA protein. And those are going to get protonated, and they're going to turn positive, and they're going to turn that protein inside out. And you're going to go through the same sort of metastable states that we saw with coronavirus, for example. And so that's going to lead to the virus opening a little pore and dropping off its genome, all 8 of those segments, into the cell. So that's the attachment and entry stage. And they don't actually split up like you can see there. That's done sort of schematically. They're going to stay together. And so it's going to be this blob that kind of looks like a sausage. And it's going to move over toward the nucleus. So the next thing the virus does is it has to find a way into the nucleus. And so this is like the Rev protein of HIV, but in reverse. The virus has proteins, and they are going to open up a nuclear pore and guide the virus in. Now, why would the virus want to go to the nucleus? Well, it's easy. Because the nucleus is full of nucleotides. And that's a great place for the flu virus to replicate. And the other neat thing is that there's no RIG-I or MDA5 or any of that intracellular immunity, no TRIMs or anything in the nucleus. Those are all just in the cytoplasm. And so for a virus like flu, if it goes into the nucleus, it can hide, and it can copy itself in peace. And then it just has to find a way to contend with the immune system once it comes out on the other side. And of course, flu has ways to do this. Okay, step 3 of the replication cycle. So we've got our virion RNA, which is going to be negative sense, so that's the darker blue. We're going to turn that into complementary RNA, neat, so that's just the opposite copy. And then from that, we're going to make both, we're going to make virion RNA again, and we're then going to export that. So the complementary RNA, some of those are also going to be messenger RNAs. So they're going to look just like a complementary RNA in terms of the way the genes are facing. The messenger RNAs are going to have a cap and a poly-A tail on the end, so just like you would expect. And yeah, these RNAs are going to be exported from the nucleus, and they can then get packaged up and we can make new viruses. So that's the next step here. We're going to assemble some new viruses, put together all the proteins that we made from the messenger RNAs, and all the brand new RNA segments that we made, and the virus is going to bud at the plasma membrane. So just like HIV did, it's going to bud from the surface of the cell and take that membrane. This is going to be different from coronavirus, which you'll remember buds into the Golgi apparatus. Corona's a weird one in that way. And then, yeah, once the virus has budded, it is ready to leave the cell. And so it does. It buds and it leaves. All right, so the hemagglutinin protein is shown right here. You can see it has a bunch of beta strands up at the top. That's those little things that look like sort of flat ribbons all in a row. And it has a lot of alpha helices. Those are the things that look like an old telephone cord down at the bottom. And this is divided up, just like all the other proteins have been, into a receptor binding top and a fusion engine, fusion motor, if you like, bottom. So the receptor binding part at the top will bind to sialic acid, and it takes 3 of these hemagglutinin proteins to make one hemagglutinin, spike, if you like, on the outside. And that's the same with coronavirus. That's also a trimer, which means 3 proteins together. And that was also true with the envelope protein of HIV. So, yeah, pretty much unrelated proteins, but they're kind of doing the same thing, which is really neat because viruses are they're basic, man. Yeah, they like what they like. Okay, so that head group up at the top, like the top of the lollipop there, is going to bind to sialic acid on who knows what protein, maybe any kind of protein. And they can actually, there's 3 molecules there, so each molecule could bind 1 sialic acid all together. How wonderful for them. And then, yeah, it's going to go through the conformational changes that lead to fusion, just like we saw with HIV envelope protein, just like we saw with coronavirus. And when I was talking about those proteins and talking about the hook-shaped fusion peptide and the conformational changes, all this stuff was originally discovered for the flu virus. And so for virologists like us, this is the OG. This is the original virus where we learn about this type of entry mechanism. And we're eventually going to get to some viruses that use a different mechanism, but I wanted to start out by highlighting a bunch of really different viruses that actually get in with a very similar sort of strategy. So, yeah, these are called type 1 fusion proteins, or class 1 fusion proteins, sorry. And, yeah, they all kind of work the same way. Pretty neat. Okay, and so this protein will have a transmembrane stalk, just like you would expect. All right, so you can block the flu virus with antibodies. Okay, we're back. The flu virus can be blocked by antibodies. Antibodies are things that are made by plasma cells in your immune system, and plasma cells are grown-up versions of B cells, so that's what you'll hear most often. The type of antibodies that block the flu are going to be called immunoglobulin type A. So we abbreviate that as IG and then a capital A, like capital I, lowercase g, capital A. And IgA is a neat one. This is actually, I believe, the last antibody that you make. So the way your immune system makes antibodies, it's going to actually start out playing around with a thing called a B cell receptor, which is like a really early version of the antibody. Then it's going to make a little 5-sided version of the antibody called IgM. Then it's going to go through some other changes through IG, let's see, D and E. And then finally you get to IgG, immunoglobulin type G. That's the main one that you would find in your bloodstream. And then if the antibody has been, proven its worth, been judged good at all these stages, then the very last stages will make IgA. And we secrete that on the mucosal membranes. So think of the inside of your nose and the inside of your lungs, those membranes. And it's great because those block the flu, and that's right where you need to block the flu. So there we go. There's some antibodies ganging up on that flu virus. Yeah, beat them up. And the antibodies don't do anything to the flu virus except for they stick to it, and they allow other proteins to come along and grab the handle at the bottom of those little Y-shaped things and consume the flu virus, or they just cover over the flu virus so they can't actually get to the cell. It just sort of bounces off, which is kind of neat. All right. So the flu has one big trick up its sleeve, and that is called antigenic shift. And so flu viruses can change in 2 ways. So antigenic drift is like normal evolution. That's where we make mistakes and whatever. Mistakes happen, get passed on. And usually the mistakes are bad, and usually the mistakes result in a dead virus. But occasionally you get a virus that's able to change and not die. And so you get this sort of slow creep from one to another. And that's slow evolution, and honestly, our vaccines can handle that. That's really no big deal. The other thing that flu viruses can do, though, is antigenic shift. And so this is going to be unique to flu viruses. HIV and coronaviruses cannot do this. So flu viruses start out with their genome broken up into 8 different segments. And when you get 2 flu viruses or more, that happen to be in the same cell, they have no real way to count up their segments, and their segments will just randomly associate, and you'll start making different random combinations of these segments. And so you can get a segment from one virus stuck onto the body of another virus, and yeah, any sort of mix and match version that you like. And that's exactly where the H1N1 swine flu of 2008 2009 came from. This was a reassortant. It had a little bit of a virus from a bird, a little bit of a virus from a pig, and a little bit of a virus from a human. And we know that pigs can get all three of those viruses, so we think it probably they came together in a pig, although we're not sure exactly which one. Yeah, hard to tell afterward. But yeah, that's where we get new flus. So new flus aren't new viruses; they're new combinations of all the parts that are out there. And yeah, birds have something like 200 different possible combinations just of the H and N proteins alone. And there's also just as many varieties of each of the other segments out there. And so, yeah, there's really a lot of ways that the flu virus can change. And so, drift is the slow, aimless drift where we build up mutations, and antigenic shift is where we swap segments around and make giant evolutionary leaps in a single bound. So yeah, that's a good thing. I want to make sure you have good notes on that. The difference between antigenic drift and antigenic shift, and their result in the result of each in sort of the pandemic potential for each. So the potential for a pandemic of just ordinary flu virus is really low because you would have antibodies from last year that would still protect you to some extent. But once you get an antigenically shifted virus, there's a potential that you've never seen this hemagglutinin before, and you have no antibodies against it. And so, yeah, that gives the virus a little head start against our immune system. So, there have been a lot of influenza A outbreaks over the years. So, there's that 1918 Spanish flu that killed at least 50 million people and probably helped to end World War I because people were dying of the flu in addition to bullets and disease and all the usual wartime stuff. Then you got your Asian flu in 1957, which was an H2N2. You got your Hong Kong flu, which was an H3N2, and this H3 was actually a modified version of the H2N2 Asian flu. It just happened to originate there. It wasn't anything specific to Hong Kong about that. 77, you had the Russian flu, H1N1, which is a descendant of the old Spanish flu. How neat. In 1997, you had a small outbreak of H5N1, which is bird flu or avian flu. Starting in 2003, you had H7N7, and 2006, H7N3 in pigs. And these are viruses that have come straight over from birds, but seem to be able to cause disease in humans. And then put all of these little pieces together, and bam, you get pandemic H1N1 2009, and yeah. A little bit of bird, a little bit of pig, a little bit of person virus, all together. And so some of these were pandemics, others were mercifully pretty well contained. All right, coming to the end of this. So let's talk the hemagglutinin protein. So the hemagglutinin protein is going to be cleaved by a protein called claritryptase. This is one of those furin-like enzymes. But the claritryptase, that's the one I want you to remember, and it's going to cut it into two parts, which are HA1, that's going to bind the receptor, and HA2, that's the fusion motor, that's going to have a transmembrane in it, and heptide repeat 1 and 2, and the fusion peptide will be right up at the front end of the HA2. And so this cleavage by claritryptase is necessary to activate the hemagglutinin so that it can start an infection. All right. And so hemagglutinin starts out as hemagglutinin 0 is what we call it before it gets cleaved into hemagglutinin 1 and hemagglutinin 2. That's just the entire protein. And yeah, that's exactly what happens. So with H5s and H7s, these really bad bird flus, they actually have a modified cleavage site. So claritryptase only needs one arginine. That's that one little R you can see right where the arrow's coming down. And that's going to be enough for cleavage. But the cleavage is kind of slow, and it does happen in the lung. Now, H5 and H7 flu viruses will tend to have modified cleavage sites that don't just have one R, they have a whole chain of R's in a row, or sometimes a lysine, which is a K as well. And once you have a whole chain of these, then there are other furin type enzymes, which are all over the place, that can now all of a sudden activate the flu. And so, with an H5 or an H7, the reason why we worry about these is because they are not restricted to the lung. They can start to grow in other body parts. And when they do, they can cause terrible immune reactions that would result in death. Whereas the normal flu is kind of limited to the lung because the enzyme that activates it is also limited to the lung. All right, so here's the hemagglutinin protein. And so we're going from conformation 1, which is over there on the left, to conformation 2. Now in conformation 2, we've lost that entire HA1 part. So the HA1 part is shown at the top there, and that's going to be the receptor binding part. That binds to the receptor, and once again, that's just to get us into the cell, get us into that clathrin-coated pit. And then once we're in there, those histidines are going to be protonated as we keep dropping the pH, and when we get to about pH 5, it's going to undergo a conformational change, and we're going to shift from the conformation on the left to the conformation on the right. And yeah, we're just showing the HA2 part. But the top bit there, that red bit, is going to have the fusion peptide attached right at the top. And the bottom bit is heptad repeat 1 in yellow and heptad repeat 2 in blue. And so they fold up together like that. And they are going to crash the cell together. So this is shown in the conformation where they've already brought the virus and the cell together. But yeah, this is exactly how it happens, and this is the actual structural proof that this is what happens. So that's kind of neat. Okay, and so last little bit is that low pH is going to be necessary, and it's going to act as a sensor that makes the M1 protein on the inside let go of the nucleoprotein and RNA to get ready for fusion. So this is going to be like the maturation process of HIV, or like what the E protein did for coronavirus. We're going to let in that little H+, the little acid ions, protons, and they are going to get in between the nucleoprotein and M1 and just cause them to let go of each other so that the virus is now ready to release. And M2 is a little tiny ion channel, so it just makes a little pore, like a little tube through the membrane, like a little Mario pipe, basically, that H+ ions can go down. Some of the early drugs that were used to treat the flu are things like amantadine and rimantadine. And these drugs worked like a little bathroom plug. They're a plug-shaped molecule. They slot in there in between those 4 helices, and they just block the H+ from going from the outside of the virion to the inside. So H+ is going to directly act on the outside of hemagglutinin and cause fusion. But unless you also activate the inside and get M1 to let go of the nucleoprotein and the RNA, this virus actually gets stuck, and it's stuck attached to that membrane at hemifusion, and it cannot go to full fusion. So yeah, by blocking the ion channel on this weird little M2 protein, we are able to block fusion from ever happening completely. So there we go. That's the end of the second section.